Hey guys, welcome to Dr. Priyanka's Biology, where we discuss everything from biology to biotechnology. So today I'm here with the part 4 of our Operon series, wherein we had seen what is an Operon, what are the components of Operon, and what is the working mechanism of Operon. So previously we had seen how the glucose Operon. Previously, we had seen how the lac-operon works. So today I am here with the part 1 of the tryptophan or the tryptophan operon. So today we will see what is this tryptophan operon or the tryptophan, what are its components and its working mechanism in presence and absence of tryptophan. So let's get started with tryptophan operon. But before we could start with tryptophan operon or the tryptophan, let's try to quickly recall what is an operon and how does it work. Whereas this operon is a very quick and wise decision-making mechanism what a prokaryotic cell like E. coli has. They are destined to understand the presence and absence of a particular biomolecule in their surrounding. So rather I would say that it is a smart mechanism which is being used by this E. coli or this prokaryotic organisms to work in presence and absence of any given biomolecule. So they are so smart because they know if a particular biomolecule is easily available in the growth medium that is being provided to them, there is no need for them to synthesize it. So that is what makes the overall gain. So if it is present in their surrounding or uh, in the growth medium that is provided to them, they will not synthesize because why spend energy? Rather, they could conserve that energy or use it for some other purpose. But when that biomolecule is not there or the organism is starving because of that particular biomolecule, so they know that's a sensor for them to understand that now it's the time for you to synthesize that particular biomolecule, maybe lactose or tryptophan. So that is how these organisms utilize this operon mechanism. So overall, if you go to see, it is a very smart move of these organisms absence of the given biomolecule. So whether it's present or it is absent, they will decide whether they have to switch on the button of that operon or whether they should switch it off. So now let's see what is this tryptophan operon. Tryptophan operon or the tryptoperon. So we have seen the background of operon. So let's see what is the definition of this tryptophan operon. The tryptophan, it is the mechanism that is involved in the regulation of the transcription of the genes which are responsible for the biosynthesis of tryptophan. So this particular operon is going to work this particular operon is the deciding factor whether the organism that is E. coli should produce or synthesis its own tryptophan or whether it should take it up from the medium. So that deciding factor is your tryptoperon. So let's quickly understand the structure of tryptoperon. Seen earlier, a typical operon structure comprises of something called as the structural gene, which encode for a single mRNA, which is going to code for the components of that particular structural genes. So in case of tryptoperon, we have 5 structural genes called as the TRPE, TRPD, TRPC, TRPB and TRPA. So like this we have 5 different structural genes which are going to code for different components or different proteins or enzymes which are required for the synthesis of tryptophan. So together they are going to channelize a pathway which is finally going to give rise to something called as tryptophan, which could be used an essential amino acid which could be utilized by the E. coli. So apart from this, we have the regulatory genes which are involved in the on and off mechanism of the operon. So here we have the structural genes that is TRPE, TRPD, TRPC, TRPB and TRPA. Then we have the promoter and the operator Now here one point we have to remember that in case of tryptophan operon, the operon region and the promoter region, they partially overlap each other. Then apart from this, yet another point that we need to remember that a repressor region has its own regulatory genes. Okay. Tryptophan operon, there is one special thing with which we have not seen in case of lac operon, that is the presence of something called as a leader peptide or the leader sequence. So eventually in further parts we will see what is the role of this leader sequence or the leader peptide. So now as we have seen the definition and the role of tryptophan operon. So tryptophan is the regulatory mechanism, or it's a regulation of the transcription of the genes which are involved or which are responsible for the biosynthesis of tryptophan. Then next we have seen that it is already comprised of the 5 structural genes. Then we know that there is an upstream promoter and operator, and together there are 5 different types of structural genes. So now let us see what are these different enzymes which are required or which are involved in tryptophan. So we know that there are 5 structural genes responsible for production of different types of enzyme. So very first one is the TRPE. This TRPE codes for or it encodes for something called as anthranilate synthase 1. Then second one, that is TRPD, it codes for anthranilate synthase 2. So they work together in coordination with each other to produce the anthranilate, a molecule called as anthranilate. Next structural gene, that is TRPC. Now, the function of this TRPC is quite different from the other ones because it is involved in 2 proteins production or the signaling of 2 proteins. So first one, it codes for and codes for 2 different types of proteins here, that is the N5-phosphoribosyl anthranilate isomerase and indole 3 glycerol phosphate synthase. Now how these 2 genes are working or how these 2 enzymes are working. Now the first one, that is phosphoribosyl anthranilate isomerase domain, okay, it first turns particular molecule, that is the anthranilate, into 1-deoxy-deribulose 5-phosphate, okay, and indole 3 glycerol phosphate synthase on the same protein. Then in turn it is going to turn the product into yet another molecule. Then the next one, that is the TRPA and TRPB, they are going to synthesize the 2 subunit of the tryptophan synthase. Okay. Together they are responsible for production of this tryptophan synthase, which is finally involved in the production of tryptophan. Now how this tryptophan happens, that is the TRPC's product are going to It is going to combine TRPC's products with serine, and which finally leads to production of tryptophan. So this pathway again we are going to discuss when we will be discussing about the biochemistry of it. But for as of now, for tryptophan, this much information is enough. There are 2 mechanisms which are involved in the tryptophan operon. So very first one is the trip repressor, and second one is the attenuation. Now as opposed to your lac operon where this attenuation was not there. Now what are these 2 different mechanisms? When we are talking about trip repressor, here we are actually stopping the mechanism or the transcription at the initiation level itself. So there is no production of any sort of mRNA whatsoever. So this is entirely regulated by trip repressor. And now how does it happen is, here the repressor is going to bind to tryptophan. Okay, and it is going to block the it is going to block the binding of RNA polymerase and further expression of the proteins. Whereas the second one, that is the attenuation, this is a special mechanism wherein it, although it is going to allow the transcription, but it is going to terminate the process immaturely or prematurely, so that whatever the mRNA is produced, it is premature or a stubby, so that it is not going to produce any sort of tryptophan, or it is not going to produce any sort of structural genes products. So now let us see the very first case. So we will learn about this attenuation in the part 2 of Tryptophan. For today we are going to focus on how does the role of repressor comes into picture. So now let's see what happens when tryptophan is absent in the medium. Because as we have seen earlier that these organisms are very smart, they know how to utilize the energy, whether they should conserve it or whether they should use it. So they are very good at taking those decisions. So example of E. coli. Suppose the E. coli senses that there is no enough of tryptophan or there is absence of tryptophan in its medium or it has stalled because tryptophan is one essential amino acid that is required for its growth. So it is sensing that it is not present. So it is going to quickly give a signal that I need to produce this tryptophan. The moment it is sensing that tryptophan is absent, what this E. coli is going to do is there is going to be a signal for this repressor. Okay, a repressor will be produced, and this repressor is inactive. This repressor, what is formed, it is inactive, and what it will do is it will try to bind to the operator, but because it is inactive, it cannot bind to the operator region, and as a result it is free. So your RNA polymerase can very easily come here, settle onto the promoter and start the transcription of the structural genes which are involved in tryptophan synthesis. So all these enzymes of the antranilate synthase 1, antranilate synthase 2, the isomerase and the synthase, all this will be produced. Tryptophan synthase B and tryptophan synthase A, which are collectively going to work together for the synthesis or the biosynthesis of tryptophan. So that is case 1. So here when there is absence of tryptophan, we know that repressor will be inactive and it is going to allow the tryptophan operon to be functional. So that is part 1 when tryptophan is absent. Now second case, when let's take when tryptophan is present in the medium. So the E. coli knows that okay, it is already present in the medium. Why should I spend my energy in synthesizing that tryptophan? So rather what it would do is, anyways, this repressor will be produced over here. So tryptophan will go and bind onto this inactive tryptophan. Let's see what happens in presence of tryptophan. So the E. coli is going to sense the presence of tryptophan in its medium or surrounding medium wherever it is growing. So quickly it knows that okay, tryptophan is already present in my surrounding area. Why should I invest my energy in biosynthesizing it? So let's use what is available to me. So this RNA inactive form of repressor will be there. So this tryptophan which is present over here, it is going to bind onto this inactive repressor. So that is going to cause structural changes or the conformational changes into the structure of this repressor, and as a result it is going to get activated, and this active combo of active repressor and tryptophan, it is going to go and bind onto the operator. and it is going to block the movement or binding of RNA polymerase. So as a result, this RNA polymerase cannot move ahead and produce or synthesize the enzymes responsible for the production of tryptophan. So that means when tryptophan is present in the medium or in the surrounding medium of E. coli, it is going to block the movement of RNA polymerase because it is already present. Why produce it? That's the simple logic which is present over here. So yet another thing, because your tryptophan is going to help the repressor in repressing or stopping the transcription of the structural genes involved in tryptophan. It is also called as a co-repressor. So the tryptophan present in the media will be called as a co-repressor. So it will be called as a co-repressor. So in this way, now we have seen two different cases, that is what happens in presence of tryptophan and what happens in absence of tryptophan. So today we have seen how is this tryptophan operon working. So there are two things, two mechanisms involved. One is when the trip repressor come into picture, and second one is attenuation, which we will see in a separate video because I don't want you all to get confused. So in presence of trip repressor, there are two things happening. Suppose if tryptophan is already present in the medium, that means the E. coli is not going to invest its energy in biosynthesizing its own tryptophan. So as a result, the RNA polymerase will be blocked by this active repressor. And how it is activated? It is by binding of a co-repressor, that is your tryptophan molecule. That is part 1. So in part 2, that is what happens when there is no tryptophan. So that means there is a signal to E. coli to produce its own tryptophan. So here it needs to block the repressor. So as the repressor is inactive, it cannot go and bind onto the operator. So as a result, the promoter is free and it can easily invite RNA polymerase to come and start the transcription process of the structural genes, which together they are going to produce, which together will work in a coordinated manner to produce tryptophan. So that's all for in this part 1 of trip operon. So I'll come back with part 2 wherein we will discuss about attenuation. So till then, keep studying and keep smiling, and like, subscribe and share my channel, and for such more videos keep following us. Thank you.